Hi everyone. We are going to try a screencast today with a computer that is not mine, and it is fairly recalcitrant. It restarts randomly every few minutes without warning, so we'll try to make through as best we can. So today we're going to talk about one of the three vegetative organs of plants. There are really only three types that we're going to discuss. Those are roots, leaves, and stems. And today we'll do roots because they are, I don't know, important, but also fairly straightforward, I hope. So the basic structure and function of roots you probably know from many years of sort of elementary school biology, but I hope I'll get to show you a few things that are sort of interesting and new. Today we're going to talk about mainly the external morphology and internal anatomy and tissue organization of roots. And then tomorrow or in the next screencast, we'll talk about nutrient absorption. And then I'll do shoots later in the week. Okay, so roots are, as I said, fairly straightforward. We think of them kind of as a column that encloses a vascular bundle or set of vascular bundles that move water and nutrients absorbed from the soil up to the rest of the plant body. And of course, roots also can help store a lot of carbohydrates for the plant as it's growing, specifically actually plants that have been storing carbohydrates for the following growing season. So two of the major functions of roots are obviously water absorption, nutrient absorption, and carbohydrate storage. I guess that's three, but there are some others as well. Other functions of roots can include anchorage into the soil, and so they can sort of hold the plant down as it extends into the air against the forces of gravity. And roots can have a bunch of other functions as well. We're learning more and more that roots and the sort of organisms that live around roots help plants communicate. And I'm using that term kind of metaphorically, but they're able to send signals and receive signals to other plants in their environments. There's a lot more to be known about that, and I'm not going to talk about it much, but it's super interesting and you should follow up on it. So the functions of roots include those and probably some other functions that we'll talk about in a moment. The basic structure and organization of roots is that there's a sort of a long portion of the root that's mostly just for transport, and that's sort of away from the tip. We're going to spend almost all of our time today talking about what goes on at the tips of the roots. So so if this is a tap root, lateral roots will emerge from the internal vascular bundles, and here's an example of an emerging lateral root. The apical meristem of the root is down here above the root cap. This is the youngest area of the root, and these are the cells that are smallest and most recently produced. They don't have much in them except for a dividing nucleus. And where the, oh my God, what just happened? Yeah. Oh my God. Okay, that was unexpected. Hello. Okay, sorry. Okay, I think we're back. Sorry. I'm using a mouse and it's crazy. Sorry about that, guys. All right, so anyway, most of our time is going to be spent here in the area of the root that's sort of really active with the soil, the area that has the root hairs, and we'll also spend some time in talking about the root cap. So this whole area here is called the rhizosphere. It is the area of each root that includes the root, the exudates or the materials that it's actually like secreting into the soil, and then all of the organisms in the soil that are interacting with it. So these include a lot of bacteria, mycorrhizal fungi, and other organisms that are sort of interacting with the roots and helping them do their thing. Okay, so we're going to spend most of our time working on root hairs and root caps, but first let's talk about a couple of different variations in overall morphology of the roots. So here are just a few black and white diagrams of various kinds of root systems. I love this one here because it's basically a diagram of some of my favorite Midwestern prairie plants that are almost all like I mean, they're near extinct in a lot of different places because we've eliminated most of the prairies. Oh, that happened again. Oh, okay. Ay. Okay, that's exciting. But what you can see from these pictures is that in some of these species, the roots are much longer and more extensive than the above ground portions of the plants, the roots and the leaves. And also the roots are, they last the whole time, whereas a lot of prairie grasses will die back at the end of each growing season. The roots keep growing at the tips, but maintaining their other structures, which is kind of cool and interesting. Here we have an orchid, and orchids have aerial roots a lot of the times because orchids will live without soil, sometimes in the branches of tropical trees. So aerial roots live in places where there is a lot of humidity in the air, and they will absorb water still, just like most of the other roots we've been talking about, but they absorb water from the air instead of from the soil. So they have different structures, and some aerial roots are also photosynthetic. So you can see if you ever look at an orchid show or go walking, if you live in the tropics and see orchids, you'll actually see that the roots are green and can photosynthesize as well as absorbing water from the air. So that's pretty cool. Here is a picture of a mangrove, and mangroves have cool adaptations which are called prop roots, and these are roots that are helping to really anchor the plant in a very marshy environment. So mangroves are aquatic plants, and they tend to live in very salty or brackish water, and they have in their roots a great system of excluding salt and basically filtering salt out of the water and allowing fresh water to move up through the rest of the plant. But they also have these cool prop roots that are really sort of easy to designate and easy to see. If you're ever down in the Florida Everglades or somewhere like that, you'll see mangroves and they look like they're walking on stilts. So they're kind of cool. All right, so here's a diagram, and I don't want to go through all of the details here except to note that at some point you should be familiar with most of these terms and be able to use them. A lot of these terms are ones that we'll talk about later in this little lecture, but here's a longitudinal section of a root, and you can sort of see the areas where they're divided into regions. So I'll talk through them from the oldest tissues down to the very youngest tissues, and then save the root cap for last. So the region of maturation is the area where all of the tissues, the ground tissue, the vascular tissue, and the epidermis are all kind of working, although the epidermis does most of its work a little bit lower down. So this is actually where the root hairs are. So these little lines here are designating the root hairs, as you can see in this little label here. The pericycle is the area of ground tissue around the vascular bundles inside the root, and it's at the center of the root where the vascular tissue is, and this is the area where xylem is moving water through the plant and phloem is moving sugars either up from the root to the rest of the plant or down from the leaves into the root for metabolism and storage. So it's at the center of the root, and remember that because it's important and it's different from stems. So it's at the center of the root where the vascular bundles are located. And it's at the root hairs where the waters are actually being absorbed into the tissues of the plant. And in the middle, between the root hairs and the pericycle and then the vascular bundles at the very center, is an area called the cortex. So the white area here is a mature ground tissue. It's mostly parenchyma cells that often will be storage parenchyma full of plastids carrying starch, but not necessarily. Sometimes they're just real thin. Excuse me. The cortex is an area of sort of stability and sort of storage for the plant, and it's made of parenchyma tissue. So, and then of course the very outer layer is the epidermal tissue, and that's where the root hairs are mature, and this is where the absorption is happening. All right, a little bit younger tissue is here in the region of cell elongation. And here what we have is the cells actually achieving their purpose in life. So the cells, once they are produced by the apical meristem at the very tip of the root, once they get a little older, they start elongating and receiving signals from their neighboring cells to develop into either sieve tubes or xylem vessel elements, or parenchyma storage cells, or root hairs, or epidermal cells, or whatever. These are the cells that have already been produced and are sort of growing and maturing and achieving, like activating their plastids and basically learning to be, I don't know, not really learning, you know this, but becoming mature plant cells. Oh my God. We're just going to deal with it and move on. All right, so that's the region of elongation. So the tissues that I talked about before are less mature. And then finally at the very tip of the root is the apical meristem, where the regions of cell division, where the cell divisions are happening. So the youngest cells are here. The quiescent center is actually probably right around here, or even lower, like right here. And these are the cells that are very, very rarely dividing. Every once in a while, they'll divide and send new, fresh cells up. And these cells here are rapidly dividing in the sort of submeristem areas, the promeristem, the procambium, and the ground meristem. So all of these areas are dividing and producing new and immature cells that then will eventually mature and achieve their functions up at this area of the rhizosphere. And then finally we have the root cap at the very tip, and the root cap is really interesting. We know it is for protection, but it has other purposes as well. Okay. Here's another example or another illustration of basically what I just said, the three different zones. The area above the tip of the root is where most of the action happens, and below that is where the cells are growing and maturing. And the quiescent center, again, is at the very tip, right above the root cap, and that's the area of cells that are very rarely dividing, and they're kind of a genetic repository, or sort of a genetic bank, where the cells are rarely dividing and rarely sort of subjecting themselves to potential mutations. All right, so I want to talk a little bit about water and nutrient uptake from the sort of external part of the from the external portion of the cell. So this is again, I'll mention it one more time. Most of the water and nutrient uptake occurs above the growing root tip. This is where the root hairs have already grown, and branch roots are sort of starting to emerge a little bit above the rhizosphere. So new, sometimes we probably should call these lateral roots, are emerging, and each of these will also have a tip similar to the primary root here. So the root hairs become extremely important because root hairs are a specialized kind of trichome, and they are able to extend the surface area of individual root epidermal cells and absorb a large amount of water, and they also can interact with the organisms in the environment, for example, mycorrhizae and rhizobium, to help with uptake of other kinds of nutrients. I want to show a very short video of root hairs. I might have shown this before, but this shows the trichome of the root hairs, and it's very short. It's only 27 seconds long, so we're going to see if this works. Okay, so here is an individual cell, and you can see the root hair emerging from the cell and maybe doubling or tripling or even quadrupling the surface area of the root, which really increases its ability to absorb water from the environment. Okay, I hope this is working. I had a little video malfunction, but it's fine. Okay, so you saw the cool root hairs. So each individual root hair is attached to a single epidermal cell. That's important thing one. Important thing two that I want you to know about sort of the root tip is that there is a root cap at the very, very tip of the plant, and those cells are also produced by the apical meristem. So every once in a while the apical meristem will divide in the other direction. So instead of making new cells that sort of contribute to the growth of the root down, the apical meristem will divide and make cells that contribute to the root cap. So the apical meristem in roots will actually divide sometimes in each direction. So think about that and how that might be different from the apical meristem of shoots. Okay, so a little bit more about root tips and root caps. Oh my God, honest to goodness. Here we go. So here's some great pictures of root caps and root tips, but focusing primarily on the root caps. So root caps, as we mentioned before, and as might be really clear to you, root caps serve the purpose of protecting the apical meristem from physical damage. So the apical meristem, again, has very little defenses because all they are basically little tiny cells that are essentially dividing or not dividing and just holding on to that genetic material. So these cells don't have protective secondary compounds or a xylem to protect them or any sort of protection except the root cap, which has thick-walled cells that produce this really great mucilage. And the mucilage has multiple functions. The mucilage is actually a complex polysaccharide, which is like mucus, so it sounds gross, and it kind of is gross. You've seen the sort of roots sort of sliming through the soil. That's because they are sort of lubricated by this polysaccharide, and the polysaccharide also serves to attract bacteria and fungi that interact with the roots in beneficial ways, although sometimes maybe not so beneficial ways too. But many of the fungi and the bacteria that interact with the roots are able to help the plant absorb materials better. Root caps also have important role in receiving, processing, and sending signals to the apical meristem to the dividing areas of the cell and also to the lengthening areas of the cell about the direction of growth and the rate of growth. So it's the root cap that somehow designates the direction and speed of the growth of individual root tips. And if you think about a grass plant that might have thousands and thousands and thousands of root caps and root tips all interacting with the shoot, those roots are really decentralized areas of sensation, signal processing, and control of the roots' growth through the soil, which helps the plants in their collection and concentration of the soil water and nutrients for the growth of the plant. So the columella cells, which are the cells of the root cap that produce the mucilage, have a lot of roles. Protection, yes. Lubrication of growth through the soil, signal receipt and transduction. And then they also occasionally will be a shed. They will be released by the root cap into the soil, into these cells that are free living from the plant. They're called border cells, and I find border cells to be one of the most fascinating things about plants because these cells are alive in the soil. They're still plant cells. They're individual cells alive in the soil, and they are still communicating with the mothership, the plant root that they come from. On the other hand, they are also interacting with the soil and sort of perceiving the soil environment, including the pH and the salinity and the water availability and other aspects of the soil. And I just don't know enough about border cells, and I need to read more about them. Anyway, I encourage you to think about how bizarre and cool that is, because the plants are essentially sending emissaries out into the world to detect their environment and then send signals back to the roots, which then can attend and change their growth patterns as a result of the signals they're receiving. I think that's awesome. All right, so now we're going to go inside the roots. This is inside the mature area, so not the root cap and not the elongation area, but this is in the zone of maturation. This is a very simple cross-section, a very generalized cross-section of the internal tissues of the plant. So remember, we have ground tissue, vascular tissue, and epidermis. Or dermal tissue. The dermal tissue is at the very outside. The epidermis of the root is the part of the plant that produces root hairs. That's not pictured here, but I'll show it to you again later. It's very, very thin, and actually the epidermis of the root, after a season of growth, the root hairs are sort of shorn off so that the area of the older part of the root no longer has root hairs, and the repair of the cells just leaves the cell without those root hair trichomes. Directly inside of the epidermis is the area that I talked about before called the cortex. This is ground tissue. It's parenchyma tissue for the most part, and it is essentially usually for storage and anchorage of the plant. These areas can also become, well, I'll just leave it there for the moment. Okay, moving inside from there, we have the endodermis. The endodermis is a protective layer of cells that we're going to talk about more in the next lecture, that sort of regulates the movement of substances from the root tips into the vascular cylinder. And I call this center here the vascular cylinder inside the procambium, or inside the endodermis, because in here what we have is the xylem at the very, very center, and then the phloem producing sieve tubes in the case of phloem, and xylem, which is producing these vessel elements, moving water all the way throughout the rest of the plant. So if you think about it, out here, if there's a root hair absorbing water, the water has to make its way through the cortex somehow, make it through the border of the endodermis, all the way through here, and then eventually enter the xylem in order to move its way into the vascular system of the plant. So remember that the water actually needs to get from the root hairs to the xylem before it is used in transpiration, and it does that through a series of actions that I'll talk about in the next lecture. So I think the other thing to notice about inside the endodermis is that there's an area of ground tissue called the pericycle. The pericycle is ground tissue, and it becomes important when we're thinking about the formation of lateral roots, or the roots that sort of spring out of the first initial primary root that the plant produces. Okay, so here's the same kind of diagram, only now it's actually a micrograph. So we have the epidermis, the very outer layer of the plant that does not have very many plastids. We have the cortex. You can see here that it's been dyed to show the starch-containing amyloplasts, or plastids containing starch. So all of these dark areas here are plastids in the ground tissue. And then the vascular cylinder is in the center. We have the endodermis. The phloem is sort of the smaller greenish looking cells in this photo here, in the middle of this vascular cylinder are these very wide bore vessel elements that are made of the xylem, that are lignified, and that are lacking cell walls at the top and the bottom of these barrel-shaped cells, and then they're stacked one on top of another so they can make a vessel for the water to flow through sort of unencumbered. So have a look at this diagram. It's in your book in chapter 24. Here's a close-up of that same vascular cylinder. So here we have pericycle cells, and those are about to be, those are about to become important. You have the phloem cells here, so you might, if you look carefully, being able to see the sieve tube elements and then the companion cells for those cells that are alive at maturity, and yet they have the sieve tubes have most of their cellular components dissolved, and there's a lot of pits between, or sieves between the cells, so that photosynthate can move easily between the cells in those living phloem sieve tubes. They're sort of regulated by the companion cells, if you'll remember, from the last chapter that we read. And then finally, the pericycle is ground tissue inside the endodermis, inside the vascular cylinder. And this is what I want to talk about next, because the pericycle becomes important when we're talking about how lateral roots form from the first primary root in the dicot root. So most vascular plants species are dicots, although some of the most common vascular plants like grasses are monocots. Dicots have the ability to form new lateral roots from within the vascular tissue of the root. So if you'll remember, we talked about the endodermis, and inside the endodermis there's some areas of ground tissue, for example, the pericycle. So the pericycle is made of parenchyma cells, or ground tissue that's sort of like the, quote, prototypical plant cell. And one of the cool things about parenchyma that we mentioned way back is that parenchyma cells can become meristematic. They can lose their function of storage or metabolism or whatever, and they can start dividing and become their own meristems and start making new tissues. And that is how lateral roots form. So here, if you look at the this area here, that is the vascular cylinder of the root, which is cut in cross section here. And what's going on here is that inside the endodermis, an area of pericycle started its own meristem and started making a new root growing sort of perpendicular to the large root that's in cross section. So this is, oh my God, cross section of a root, and it's got a new root forming out of here. So it has a new apical meristem kind of growing sideways. It has new areas where these areas are going to eventually become grow root hairs and have vascular bundles that are, look at this though, the cool thing about it is that, oh my God, all right, stop, holy moly, all right. All right, I'm going to be very careful. I get excited. All right, so here what we have is the lateral roots. What I really want you to notice about this is that the lateral root vascular system is physically directly attached to the vascular system of the primary root that it was that it's growing from. And that's true of this younger one too. You can see this is the vascular system. Here were the original meristematic cells that kind of sort of awoke and started dividing and making new roots in sort of a perpendicular fashion to the original root. Oh, okay, that was a long explanation. Here's a monocot root. I'm not going to go into this in too much detail except to show you that the vascular bundles have a slightly different organization. I'm not going to worry too much, but you also have vascular bundles and new roots forming from the monocot as well. Again, from the parenchyma storage tissue, which has received the signal, the sort of hormonal signal from the roots to form lateral roots to become meristematic and start dividing and make new organs, making new roots forming perpendicularly as lateral roots. Okay, I think that is pretty much where I'm going to stop, because we're going to pick up in the next lecture with the Casparian strip, which is the area in the endodermis that allows plants to regulate their transport of water. So thanks, and I'm sorry for the crazy computer, but at least it didn't crash. All right, hope you guys are doing well, and we'll talk to you later.